Hello everyone, myself SAKSHI Shamla, and I have completed my master from Delhi Technology University in biotechnology, and my topic for today assignment is the identification of plasma protein signature which are associated with the progression of the cardiac gastric cancer and the precancer lesions. So first we are going to dig on the details of the corresponding author of this papers, and they are Xiaoming Wing and Wing Wing are the one which are affiliated at the Office of National Center Cancer Registry at the National Cancer Center in Beijing, which is in China, and the area of their research which is generally focused on the cancer epidemiology and which include the survival and the certainty of the trend in cancer and the cancer incidence as well. And the major role which they have played here is the collection of cancer data nationally, and have done analysis that also involving the strategies which can be used for the prevention, and thus they are focusing on providing a good for improve public health outcome and cancer management as well. So these are the details of the corresponding authors. Now we're going to look on the detail of the background of this literature. So in this case, the progression of this cardiac gastric cancer, which is CGC, this is often preceded by a precancerous lesion, and this is very crucial for us for its effective management. And thus, as from now, we have seen there is a limited biomarker or potential novel protein for its targeting process, and thus this study is utilized where we have used plasma protein which can provide a distinct nature which can be associated with CGC. And for this study we have highlighted 162 164 negatively associated and 42 positively associated protein for identify the target potential for this condition, which is cardiac gastric cancer, which have a very high diagnostic accuracy. Thus it can be used to differentiate the different stages of the gastric cancer, and thus it provides us a better understanding of the molecule signature as well. Moving on to the introduction part. So in the introduction, as the cardiac gastric cancer is a subtype of the gastric cancer. So it poses a significant challenge, significant health challenge, as shown here in this figure. What are the difficulties which are faced in this condition? So one is they does not show any symptoms, so they are asymptomatic. Thus there is a late diagnosis, result of which, and the diagnosis required endoscopic bioscopies, which are invasive and not suitable for a large scale process. Additionally, existing serum biomarker lack the sensitivity and specificity which is recent in the advancement in proteomics. And if you see the technique which is used here in this paper, which is the data independent acquisition DIA, as shown here in the figure, are the one which offers us to identify a new non-invasive biomarker. So in this, the objective is in this study, which is to aim us to uncover the plasma proteomic signature that can differentiate between the disease condition from its precancerous lesions and the normal one, and thus empower us early detection. This can be used for the strategies in gastric cancer. Moving on to the next, which is the methodology. So in this we have used 4 samples. One is the normal, which is given here in the figure. If you can see, this is showing us the flow chart of how the process they have proceeded for this experimentation in this literature. And first, the blue ones in the normal, and this CLGD, which is given in yellow, are the cardiac loop grade dysplasia, and this CHGD are the one which is called as cardiac high grade dysplasia, and CG CG is the one which is the gastric cardio cancer, simple one, and we have taken almost 40 samples for this. And then we have divided the sample into two parts. One is going for the DA, which is the data independent acquisition, and another is going for the DDA, which is data dependent acquisition. The difference is there in, if you seen in clearly in the diagram, which is DIA goes for the separated sample separation, which is the 4 different sample are separated and then goes for the other treatment, whereas in case of DDA, the sample is mixed and then their protein extraction, which is done by reduction, acylation, and thus dilatation is performed. So for the identification of the biomarkers, we have used LC mass spectrometry, which is a potential platform for identifying the biomarkers, and this is how we have done this. And from this DDA we have done this data search, and then spectral light, which can further convert into DI for quantifying the proteomics data. Next, this is the this flowchart is the one which is showing us the details regarding the bioinformatic flow work. So in this, the plasma plasma samples which is we have taken, which is n number of 40, and we have then quantify using this method, which is DIA, which is the data independent acquisition. So they are the first we done this PCA, which is principal component analysis, and this PLCTA, then form a volcano plot. Also we have done in this case, this is random forest method, from which we have utilized from which we have identified the top 20 protein, and this VGCNA, which is the weighted gene co-expression network analysis. So this is used to identify the positively associated and negatively associated protein. So the one which is the forest, which is given in the blue, and the one which is given in the orange color, which is WGCNE, have taken, and then we have drawn a Venn diagram, and from them we have chosen the intersection protein, which is given for the further processing step, where we have used this LSSA regression for further finding out the specific data and compress the data which is given earlier to us. And then we have done a potential protein marker panel analysis, and finally we have performed some methods in order to identify the specific protein for the study. Moving on, some new technique and instrumentation which are used in this literature. One is the data independent acquisition, which is I'm repeatedly using this term, as this combines the protein wide coverage, which is not seen in DNA, with reproducibility and accuracy, good sensitivity, which can be a targeted method. Then we have a PCA. PCA is the one which is the principal component, as I told. It is the one which is used for the unsupervised, or we can say simply the reduction. And then we have this principal. To complement this principal component, we have used this partial least sequential discriminator analysis, which is PLSADA. So this provides us a supervised analysis using this technique, from which we can define the protein to be up or down based on their fold, log fold to ranges changes. And then we have this VICA, which is VGCNA. So this is used by our version or R package, and this provides us the correlation between the disease and the module courses. And then we have this random forest. So random forest is also a model that constructs the a kind of a classifier which can be used to differentiate the different group, with the four different groups which we have seen here. And this the one with last one, which is the least absolute shrinkage selection, as the name itself suggests, shrinkage selection and operator. So it is a regression which is imperative for further variable selection and the shrinkage as well. So moving on to the results. So as we have performed this DA with the targeted data which we have extracted using the sample extraction from the 4 samples in the starting process, and using corresponding DDA run which we have done. Now the result from the PCA, which is shown here in this diagram as well. This PCA component result, second one shown here by the black arrow, this shows the variability, like this PCA saw a substantial variability in the distribution of proteins in abundance. And for this PLCA, which is complemented with this, so in this the samples are primarily clustered by a group, and to further visualize this data we have used a volcano plot in which we have uses all the conditions to differentiate between each other. So in this diagram I'm only showing this disease condition with the healthy one, and then we have performed this weighted gene co-expression network analysis where we have bounded the 7 modules, in which the green and blue are the one which is given for the enrichment analysis, where we have performed gene enrichment analysis and get pathway analysis. Then we perform this, as I told, the reform the random products module. Also, after which we, as I told in the step, the one here, that we have chosen the Venn diagram from this random forest and Vega. So this step is also showing the same process, but in diagrammatic Venn diagram format, where 200 of random forest classification were used and significant module of 207, among which 67 were the common between both of them by using the Venn diagram. And then we go for the further reduction by LSSA, and after reduction, we move for the protein identification step. So here the protein identification step, there are 3 proteins which are upregulated and 2 proteins which are downregulated. So if you see here in this case, the red one are the cases which is the disease condition taken, and the green or this blue-green shade is given as the normal one. If you see here in the LY6G6F, so this is showing this red one is the case one, and the blue this blue-green one is the normal. Thus, if it is lower than the normal case, then then it is down-regulated. So it is down-regulated in this condition. Similarly, this is how this protein expression is accounted. Then we move for IHC by which is immuno this IHC, and where we have performed by IHBA, which is shown as this 3 proteins which are then confirmed as a result. And finally, to further validate this, we have performed this RO analysis, which is shown as the higher accuracy using this EOC for this 3 specifically protein. So this then can be used for the separation of classes by this method, which is PLCDN into 4 groups. So moving on to the result, this like as I have said, we can differentiate between the 4 groups of the disease condition, we have identified the 3 novel proteins. Therefore, the protein identification has been done, and we have uses the technique which is random forest and also regression and immunohistochemical result, which is from the HPA data basis. Moving on to the conclusion point, so we have identified the potential novel markers for this condition, which is cardio gastric cancer, and this can be used for high population rates for the precancerous lesion as well. So the key finding of this paper are the testing proteomic profile, as this can be used to differentiate between the 4 groups as I told, which are the high grade dysplasia, low grade dysplasia, and the cardiac gastric layers, and from the healthy controls. Then we have and also we can use this for the cleaning application, for as we have identified it as a biomarker used for early detection and further validation as well. And for candidate protein, the 3 protein are found out with which is demonstrate a very high diagnostic accuracy with the AU values of 0.91 for CLGD and 0.99 for CHGHD, and others except are 0.98 for the CGC. So moving on to the next is the, so what we learned from this manuscript are that we can develop a mechanism or method from which we can identify a technique which is a non-invasive biomarker, and this is specific for the plasma protein to serve as a marker for this condition, which is cardiac gastric cancer. And we can also this also provide us for the distinction between the 4 different grades which I have discussed, like high grade, low grade dysplasia control, and the condition of cardiac gastric cancer, and as also the 3 continued identified, which is highlight the diagnostic accuracy as well. So moving on to the next, which is here given in the future plan. So what we can do for this in the future, so we can provide this for the larger scale validation study and conduct a larger multi-center study for the further validity and diagnostic performance to identify biomarkers panel and clinical utility for the biomarker. And I am repeating this thing, like it can be used for early detection and the risk stratification for the condition of the precancer lesion, and for the it can be used for mechanistic studies. So in this we can investigate the fundamental roles of the specific 3 proteins which we have find out for their better understanding in the therapeutics role, and we can also understand its molecular pathway and the network which can be associated with this identified protein. Thus we can gain insight into this biology and the progression, and this longitudinal molecule where we can evaluate the changes in the biomarker over a period of time in the patients which have this condition with the precancerous lesions, to aid, thus we can able to identify potential predictory progress in this condition, and thus we can assess the utility of this biomarker and for monitoring the treatment and responses of the disease if there is a reoccurrence occur. So that's all from my side. Thank you.